So it's a pleasure to introduce my friend and colleague, Guoping Feng. Guoping is the James and Patricia Poitras Professor of Neuroscience in BCS and the Associate Director of the McGovern Institute. He's an institute member of the Broad Institute and the Director of Model Systems and Neurobiology at the Stanley Center. Guoping received his medical degree from Zhejiang University, one of the top universities in China, and then came to the US where he received his PhD in molecular genetics from SUNY Buffalo. He did a postdoc with Josh Sains at Washington University and then joined the faculty of Duke University in 2000. We were extremely fortunate to recruit him to the McGovern and BCS faculty in 2010. Guoping's received many awards. Last year, he was elected to the American Academy of Arts and Sciences. He won the Scientific Innovations Award of the Brain Research Foundation, the Gil Young Investigator Award, the McKnight Technological Innovation Awards, and many other awards I won't have time to list. Now at MIT, we often say that our best translational research is done by basic scientists who are keeping an eye out for potential human applications of the discoveries they make in the labs. And Guoping is really the prototype for that type of scientist. Early in his career, he conducted molecular genetic analyses of several important synaptic proteins and created many molecular tools and modified animals in use by labs around the world. While he was doing that work, it was becoming apparent that the vulnerability genes for many psychiatric disorders impact synaptic proteins. So Guoping saw an opportunity to create new genetic models for both understanding the impact of these mutations on synaptic and circuit function and building the conceptual foundation for the molecular therapies of the future. He's widely recognized for his work on SAPAP3 and SHANK3 in mice, which are scaffolding proteins that seem to play an important role in autism, OCD, and schizophrenia. Over the past 5 years, he's also become one of the world's leaders in creating new primate genetic models for psychiatric disorders, including the first transgenic marmoset program here at MIT. I should also mention he's recognized as one of the most generous neuroscientists in the world, making his tools and genetically modified animals available to labs all over the world. Now today he'll be talking about some of his basic research on the thalamic nucleus of the thalamus, which not surprisingly are leading him in new therapeutic directions as well. Guoping, I turn it over to you.
Thank you, Bob, for the very nice introduction. And thank you everyone for attending the seminar. Today, I'm gonna start to share my screen first before I start talk. Can you see? Okay, great. So today I'm gonna talk about some of the new recent studies in my lab focusing on thalamic reticular nucleus, their function and its function and particularly its potential dysfunction in neurodevelopmental disorders. So my lab is very interested in synapse and circuits development, and particularly their dysfunction or defect in neurodevelopment and psychiatric disorders. So most of our work is focused on one particular circuit called corticostriatal thalamocortical circuits for two reasons. One is for many of us use animal models, we're looking for circuits that are relatively conserved across species. The second is this circuit's dysfunction of this circuit is associated with many of the key symptoms in psychiatric and neurodevelopmental disorders, including repetitive behaviors, cognitive inflexibility, sensory abnormality, and sleep disruptions. Just name a few. And also many of the studies have implicated dysfunction of the corticostriatal thalamocortical circuits in ASD, OCD, ADHD, or schizophrenia. So previous work in my lab had been focused on corticostriatal synapses in repetitive behaviors. And more recently, we focused a lot of attention on thalamocortical circuits, on sensory processing, attention, sleep. So this is the part I'm going to talk about. So today I'm going to focus on one particular part of the thalamus called the thalamic reticular nucleus, TRN. You probably all know very well TRN is a group of GABAergic neurons that provide a major source of inhibition to the thalamus. And the main function of TRN is like we call the gatekeepers. Basically, it selects relevant information and suppress the distractive information, distractors. So during sensory gating, attention, or cognition. And one of the best example people use for the TRN function is if you are in a very big party, you know, we don't have that right now, but it will resume sometime, I hope. And they're very noisy, but you can still focus your attention to your friend who have a very nice conversation. So how do you filter all this noise out? The TRN play a very important role. That's just one of the examples. And TRN also play a very important role in generating of sleep signals. So they play a very important role in regulating sleep. And also importantly, TRN dysfunction have been implicated in many of the psychiatric disorders, including autism and schizophrenia. So I'm going to separate today's talk into 3 parts. One is the first part I will use the patch D1 as a neurodevelopment disorder model in mice, and talk about how dysfunction of TRN may contribute to some of the key symptoms, key phenotypes, including attention deficit and sleep disruption. And in the second part, I will talk about our very recent work using integrated single cell analysis and to identify distinct TRN subnetworks, how they may control different aspects of TRN function and help us to understand the brain disorders. And in the third part, final part, I will talk about a very new tool we developed that can allow us to perturb or probe circuit function and dysfunction. A highly sensitive optogenetic tool allows to non-invasively or minimum invasively doing optogenetics. So one of the reasons that we are interested in TRN is that we actually realize that many of the risk genes for schizophrenia, ASD, are highly expressed in the TRN. If you, I just give a few examples. In schizophrenia, calcium channel CAV3.3 and the mGlu3 are both GWAS hit risk genes. You can see they're very clearly highly expressed in the striatum, the TRN in the thalamus. And similarly for some of the genes called monogenic disorders in autism spectrum disorder, neurodevelopment disorder. Today I'm going to focus on patch D1. And so what is patch D1? So first, before I start talking, I want to acknowledge people, the key people who actually made a major contribution to this work. This was led by 2 people. One is Mike Wiles, my former graduate student, and also very closely working with my classes lab, Ralph Wimmer, Mike's postdoc. Both of them contributed very significantly to this work. So what is patch D1? So patch D1 is a 12 transmembrane protein. Actually, the function is unknown. It's X-linked. In humans, deletion of patch D1 in boys lead to very severe neurodevelopmental disorder with intellectual disability. And they have many symptoms. They range from sleep disorders, ADHD, muscle problem, hypotonia, and also aggression and ASD. And of course, most significantly, they are intellectual disabilities. And the clinical study suggests that deletion of this gene may account for close to 1% of patients with intellectual disability and autism spectrum disorders. So we decided to look at how, decide to make a knockout mouse to study whether the defect, how this defect, deletion of the gene affected the TRN function, because we found that early on during the early postnatal days, patch D1 is almost exclusively, very highly expressed, enriched in the TRN. Later on, this is a picture shows the P15, that later on, it's still highly enriched in the TRN, but it started to express in many other brain regions, including striatum and the cortex. So we made a knockout. We collaborated with my classes lab together, using in vivo recordings show that indeed in the knockout mice, they have reduced bursting activity in the TRN, and in the cortex you can detect the reduced sleep spindles. So that suggests that they may have disrupted sleep. Indeed, when we use behavior test to look at the sleep, these mice did show increased number of sleep bouts. That means their sleep bouts is shorter, and they are suggesting they wake up many more times during the sleep. So Actually the TRN bursting activity has been very well studied. There are two types of channels that play a critical role. One is CAV3.3 T-type calcium channel. The other is SK2, which is a calcium activated potassium channel. And previous work have shown if you delete either one of them, you will lead to reduce the bursting and reduce the sleep spindle. So we decided to in slice to look at whether we have reduced the bursting activity and whether we can detect the defect in either or both channels. So indeed, when we Ralph did the slice physiology show, indeed in these animal, in the TRN neurons, they have reduced the bursting. And however, we did not see the calcium channel defect. The T-type current is normal. We did see a reduced SK current, suggesting that the defect actually is in the SK current. So because the SK is SK2 channel is calcium activated potassium channel, so we look at whether this defect is coming from a calcium homeostasis defect. So in slice, Mike and Ralph worked together to indeed show that in the knockout the neurons, we see impaired calcium homeostasis. The calcium concentration is much lower. So we hypothesize that this defect may be the cause of the TRN dysfunction. I will show you a little bit of evidence later. So as I mentioned, the TRN play a very important role in filtering distractors. That means they are very important for attention. So Mike's lab actually developed a very nice assay, mouse behavior assay, to study how to study whether these mice actually have attention deficit like behavior. So the assay is basically you train the mice to anticipate, to learn that visual cue, and once they learn the visual cue, when the right cue showed up, mice will know and will get a reward. They can poke and get a reward. And if they did it wrong, then they will have a timeout and no reward. So in both knockout and wild-type mice can be trained very well. The accuracy reaches about 70 to 75%. And then during this anticipation before the presentation, we can also add a distractor, a different cue, visual cue or audio cue. And the wild-type mice continue to doing well, but the knockout in this case with a distractor actually make many more mistakes. So suggesting that there is a difficult for the knockout mice to fill out these distractors. And so we think this is attention type of behavior. So in addition, if you put a mouse in an open field test, the mice actually, you can see for the bottom of this figure, they're constantly hyperactive. So we think this put them together, we call them ADHD-like behaviors. So, but in addition to the sleeping problem, attention, and hyperactivity, these mice actually show many other problems, including learning disability in fear conditioning and in inhibitor avoidance test. They are very aggressive. They also have motor problem, muscle problems. So the question is, which part of these behavior are coming from the defect in the TRN? Because early on, patch-doing is enriched in TRN, but later on they're also expressing in many other brain regions. So we found a way actually to selectively delete the patch-doing in the TRN. The way we did it was to get an idea from Bo Lee's group at Cold Spring Harbor. We combined patch-doing, conditional patch-doing mice with the somatostatin Cree. In the TRN, both are inhibiting neurons, both patch D1 and somatostatin Cree are highly expressed in the TRN, but outside the TRN, they are actually not very little overlap. So we actually specifically made a patch D1 YP mice to show that there are very good overlap in the TRN, but very little overlap outside TRN. So you combine these two mouse together, you get a conditional knockout, the selectively knockout patch D1 in the TRN. You can see this is the control in the TRN expression. This is in situ, and you can see in the knockout, conditional knockout, only the TRN patch D1 is deleted, but the rest of the brain is still have patch D1. So in this selective knockout, we only see attention, we still see attention deficit, we see hyperactivity, we see sleeping problem, but we did not see aggression or learning disability or motor problems, suggesting the TRN defect indeed is responsible for the hyperactivity, attention deficit, and sleep disruption. Now, as I mentioned that we think the SK2 channel defect, current defect in the TRN play a key role in this TRN dysfunction. So luckily, there is a compound called 1-EBIO, which is the SK2 activator. So we actually tested whether this drug, by activating the SK2 channel, we can rescue the defect. So indeed, when we inject the 1-EBIO to the knockout mice, we can rescue the defect in the TRN inhibition to the thalamus through the chloride channel imaging. But more importantly, it can also rescue behavior. It can rescue hyperactivity, it can rescue attention deficit, but it cannot rescue other problems like learning problems, motor problems. So suggesting indeed the SK2 channel defect in the TRN we identified before is the cause of this type of behavior. So we all know that sleep disruption, ADHD, sensory abnormality, which are core, some of the core functions of TRN, they are very common in neurodevelopmental disorders, in like autism spectrum disorders. But these are very diverse functions, right? So this type of TRN dysfunction contributing to neurodevelopmental disorder also led us to think about how the TRN, such a uniformed inhibited group of inhibited neurons, how do they control such a diverse function? So we start to think about whether they have, whether all these functions are controlled or mediated by same circuits, are they a cell type or circuit heterogeneity in the TRN? Can we identify TRN specific target or even subnetwork type specific target allows to more precisely modulate the TRN function, and hopefully in the future identify targets to develop treatment for some of the symptoms. So for this type, in my second part, I'm going to talk about how our recent study, we use single cell analysis to identify distinct subnetworks in the TRN. This helps us to think very differently how can we modulate TRN function. Again, I want to acknowledge the people who did this work first, and this work was led by 3 key people in both in my group, in Zhang Yanfu's group at the Broad, and also in Joshua Levine's group at the Broad Institute. Ying Qing was a postdoc fellow. He is now assistant professor at Tsinghua University. Violeta was a postdoc also, and she's an assistant professor at National University of Mexico. And Shen is a very talented research associate at the Joshua Levine's group. So TRN, you can see, is a very small portion of the brain, but if we want to do single cell, we need a way to get them out, right, specifically. And we found that at least in mouse, almost all the TRN neurons are PV positive. So that allows us to make, PVTD tomato mice and label the neurons with, red fluorescent protein. Then in the section, you can cut this in the fluorescent dissecting scope. You can cut the TRN out and doing single nuclear RNA sequence. So with this approach, we actually sequenced the, many neurons and did the data analysis. What we found is it's different from many other brain regions. We actually couldn't find 2 or 3 or any distinct cell types. What we found is there are 2 groups of cells that their gene expression form a gradient. And each group has hundreds of genes expressing a gradient. So one group, if you select these genes that form a gradient in this group, then the other group will have the opposite thing, which means that the gene expressed highly in the first group would be expressed lower in the other group. So you can see from the figure, this figure on the right side, you can see there are 2 groups of each actually row is a gene. You can see their gradient expression in different cells. So some of these genes are very selective. There's almost or minimum overlapping that allows to use this as a marker. So we pick the two markers. One is the SPP1, you can see on the top, and that their expression gradient pattern is almost non-overlapping with the other group. We pick the ECEL1 as a marker. So use this two marker now allow us to identify these two cell groups in situ. This is, mixed all the cells together. Now, how do they look in the real brain, right? After you get the single cell RNA data. Because of these two markers, now we can do in situ hybridization, use a fish to understand what are these cells, what do they look like, how they are connected to the other part of the brain. So what we found is that with in situ, we found very surprisingly that these two group of cells, they are anatomically distinct. They are actually segregated. So as I mentioned, all the TRN neurons are PV positive. So the red color is albumin antibodies in situ. You can see that they are red, but all the SPP1 cells seems like in the middle, and the ECEL1 cells are actually on the edge. So they form kind of shell and a core structure in the TRN, although TRN is a very thin layer of structure, but they still have a very distinct anatomical differences. Because these cells have differential gene expression, you can predict they must be electrophysiological property defect, because all the few of genes we showed here are ion channels and neurotransmitter receptors. So based on this kind of feature, we decide to looking at recording from each type of cells. The way we did it is to use called patch-seq. Basically, you identify the cell, you patch the cell, you record. After you recording, you take the nucleus out and doing single nuclear analysis sequencing to look at what is this neuron you have just recorded. So what we found, indeed, they are very different. The SPP1 positive cell, which is in the core part of the TRN, they usually have multiple bursting, and within the bursting they have higher frequency. On the other hand, the ECEL1 cell, they have very few bursts, usually 1 or 0. Right? And within this bursting, they also the frequency, 5 frequency are much lower. So that means that not only this is actually consistent with their expression, as I mentioned, the T-type calcium channel and SK2 channel play a very important role in the bursting, and actually in the SPP1 cell, they have a high expression of this type of channels control the bursting. And so not only not only these cell types has the molecular distinction, but they also have cellular property differences. But in the thalamus, they actually have many different type of subnuclei, right? So generally people can separate it into two large groups. One we call the first order thalamic nuclei. These are the thalamic nuclei directly receive input from peripheral sensory. So they call the relay neurons. They basically relay the sensory information from peripheral to the cortex. Then there's another group of thalamic nuclei we call higher order nuclei. They basically they don't receive directly from peripheral sensory. They receive and input from different cortical areas. So they more likely play a role in the cortical in coordinated cortical interactions. So both all these thalamic neurons actually receive TRN input. So what our question is whether they receive the same TRN neuron input or they actually have a different circuits and different group of TRN neurons. Now with this type of subcellular specificity, now we can actually answer this question. The way we can do it is we can use a retro bead, inject into either the first order thalamic nuclei or high order thalamic nuclei, and then doing in situ to look at double in situ to look at whether this tracing matches the SPP1 cell or matches the ECE1 cell. Right? So we can do many different injections. What we found it was also very surprising that if we inject the retro beads, which is red, into the first order thalamic nuclei, then look at the staining in the TRN. What we found is they're always in the middle. It matches the SPP1 expression. If you inject into the higher order thalamic nuclei, these retrograde beads goes to the edge, which matches the ECA1 cell type. So you can do this kind of for multiple, sensory modalities, including auditory, visual, and somatosensory. We did all three of them. And what we found is indeed, they have a very distinct connecting patterns. That means for the first order, vast majority of the first order neurons in these thalamic nuclei receive SPP1 positive TRN neuron innervation. On the other hand, for the high order thalamic nuclei, whether it's somatosensory, auditory, or visual, the vast majority of them receive SECA1 positive cell projections from TRN. So that all this suggests that not only in the TRN, not only we have molecular heterogeneity, that you have two groups of gradient expression cells, you have these two cells, we also have cellular function heterogeneity. But most importantly, these molecular and cellular heterogeneity actually are connected with circuits specificity. That means there are two subnetworks in the TRN that can control potentially different function. So now the question is, why do you need these two? How do they function differently in the intact system? How do you control behavior use this? So we actually did two things. I'm not going to have time to go through all of them, but I can mention briefly. We did actually in vivo CRISPR screen, because we have all these gene lists. Combined the pooled CRISPR screen, identified actually several genes are critical or very important for the bursting activity, in addition to the CAV3.3 and SK2 channels. And this is for the in vivo CRISPR screen for molecular mechanisms, but it also allows us now to do look at in vivo function and behavior. We actually selectively perturbed the function of the SPP1 cells or ECA1 cells. We found that SPP1 cell play a very important role in mediated sleep. So, but the next step really is to generate genetic tools allow us to much more precisely control the function of each group of neurons and really figure out more systematically what each subset network can do and how they might be affected in the neurodevelopmental disorder models. So we now just showing some very preliminary studies. This was done by a postdoc, Sasha Crow in the lab. She made the TRN Cree lines and made the ECA1 Cree on the shell like on the edge of the TRN. And also she also made the SPP1 Cree, which is the core part of the TRN. So we hope these tools will allow us to really dissect the subnetworks of the TRN function, and we hope use this type of tools to really dissecting how the TRN subnetwork may be affected in different developmental models, mouse models of neurodevelopmental disorders. So, and finally, in the last about 10 minutes, I'm going to talk about a new tool. So we always try to, since we are studying circuits, we always try to develop new tools that facilitate our study of dissecting our circuits function and the dysfunction. And today I'm going to talk about a very new tool we just developed. Actually, this paper just came out in Neuron a few weeks ago, and we called SOUL. A new step function option with ultra light sensitivity. This again, I want to acknowledge people who played a key role in this study. Xin Gong actually is, oh, sorry, I misspelled the name. There's an extra G, there's an XIN. Xin Gong, she was a former graduate student, and Jonathan Ting was a former postdoc fellow in my lab. We collaborate with Bob Desmond's group, and Diego is a postdoc in Bob's lab, and we worked very closely together to characterize this new tool. So you're probably very, very familiar with variety type of optogenetic tools. The original optogenetic tools, Carl Dadros, Boyden, Feng developed was called channel rhodopsin. And later on, the second generation is called step function opsins. The difference is channel rhodopsin is a very high, fast action and give you very precise control fire frequency, right? This is from one of our early studies showing the cell type specific mice. You can, whatever the pulse, this little, spike, and they basically, as soon as the light on, channel will open, right, and can generate action potentials. Light off, it will close, and so you can precise the control, whether it's 5 hertz, 10 hertz, 20 hertz, up to 100 hertz with some of the modified channel opsins. So this gives you precise, fast action. And then there are second generation, which is called stable step function opsins. The difference is it's cumulative. When it's open, it's gradually open and cumulative, and so it's slower. But the unique feature is it's a very prolonged action. So this is like, this is probably 5 or 10 seconds of light pulse, then channel opens. This is current, photocurrent. You can channel opens. And another unique feature is you can actually have a different wavelength. It's 590 yellow light, actually turn it off. So so you can see that there's almost like 30 minutes, half life. So that means you can do a lot of things with complex behavior. You don't need the optic fiber stick to the head, and then you can do. So we thought this might be for some of our circuit analysis behavior, this might be much more useful. So we start to experimenting with this type. What we found is it's the original SS4 was not really as sensitive as we hoped. So our original goal was that can we make some change and make it more sensitive so it can activate a large part of the brain and doing behavior analysis use the SS4 instead of channel rhodopsin. So what we found is that if there were a lot of previous studies by many groups which contributed to the development of improvement of optogenetics. There's one particular mutation, T159C, and which is when it's added to channel rhodopsin, it can dramatically increase photocurrent. We thought this might be a good way to increase the photocurrent of the SSFOs that make it more can be more useful for our study. So we indeed added this mutation to the SS4. We found indeed it significantly increased the photocurrent. So this is in cultured neurons. You can see with the same light power, the soul has a much more bigger current. And in the mouse brain slice, the similar thing. You can use a pulse of blue light to activate, pulse of orange light to inactivate, right? And you can see, and not only increase photocurrent, but also the importance is maintain the feature of the SS4, which means you can activate it with blue light, inactivate with orange light, and it lasts a very long time when you activate it, right? So it's a similar, the tau is very similar in this SF4 and in soul. So they give us a hope that this may be more useful for controlled neuronal activities to doing complex behavior. That's our original goal. What we found was very surprising. So we were not, so what we found is whenever we try to do surgery, open a skull doing mouse surgery, put an optic fiber in, this neuron already firing in the surgery light, not the blue light, not a laser. So that at the beginning we were disappointed. We said, oh, this is really too sensitive, and then we cannot use it for it because the mice, when you do surgery, you already activate a neuron. Then we realized maybe if it's so sensitive, maybe we should try outside the skull without surgery. Maybe you can the light, enough light to penetrate to activate the neurons. So that's how we end up try to test. Indeed, what we found is that whenever we put lights outside the skull, we can activate the neurons inside the skull. So that's what we call non-invasive transcranial activation in mice. So we worked again with my Classa's lab, and we injected this SAW. We did a comparison. One side we inject SS4, the other side we inject the SAW. Then we put electrode in, then we shed lights from outside the skull, right? This is in the dorsal medial thalamus, which is midway of the mouse brain. And what we found, you can see is that you can activate it with blue light in the cell and inactivate after the orange light. And you can see there, with this is actually quite a small, light power. You can see significant activation, and with this small light power light, actually you don't activate the soul from outside skull. So this gives us hope that maybe you don't need optical fiber sticking to the brain to do optogenetics anymore into the mouse brain. So the question is how deep you can go. So we decided to test the deepest part of the mouse brain, which is one of them is the lateral hypothalamus. And we also want to choose an area which we can clearly demonstrate the behavior. So this is a well-studied area for feeding. So we inject the soul P2A-TD tomatoes, so we know where they are expressed, how they are expressed, into the lateral hypothalamus. And you can see, we can do, then you shed lights from the top of the skull and without any optic fiber inside the skull. And you can see that if you do false staining, you can see with SAW, you can see that there are many neurons activated, right? So M-Cherry we use as a control virus. So you can see the dramatic increase in the activation of activated neurons measured by C-force expression. So then we can do, because this has been really well studied, people have using channel rhodopsin to activate the use chemokines to positive neurons. You can suppress the feeding. So the question is, can we use the light from outside the skull suppress feeding? So you basically food deprive the mice for overnight. Then next day you basically give the food, mice will eat, right? So so this is the control. But when you turn the lights on from outside the skull, right, for a few seconds, then you stop light, you measure the food for 10 minutes. Food intake for 10 minutes, you can see it dramatically suppress the food intake from these mice. Then you can, after 10 minutes, you can turn the orange lights on. Then you can turn the lights off or turn orange lights on, and you can see that the animals actually restore feeding. So that is suggesting that not only you, from transcranial, not only you can activate neurons in the one of the deepest part of the mouse brain, but you can also change the behavior. So since this is, so is a step function option, you actually can take the optic fiber off, then you can allow you to do much more complex behavior testing. So, and one of the things, the transcranial activation can avoid the brain damage, right? So inflammation. So actually, Xing and Diego did an experiment to show that indeed put optic fiber in, you cause a lot of inflammation. This is measured by IBO1 activated microglia. You can see this no treatment, transcranial light stimulation, and this is optic fiber sticking in, right? This is a contralateral side. You can see there's almost no difference between the transcranial light stimulation with the no treatment. So suggesting that this approach may be in some of the cases the much better approach for study the brain function and the dysfunction. So one of the disadvantages of the optical fiber is that in mouse, the brain is so small, and 200 micron optic fiber light source enough to activate a big proportion, a percentage of brain structure. But for bigger brain, like macaque monkeys, that has been really difficult to generate really robust behaviors because the optic fiber is so small. You put a very big one, then your brain damage is too big. So we want to test whether this sensitive, super sensitive opsin can allow us to do optogenetic activation in macaque monkey brain without stick optic fiber into the brain tissue. So we cannot do the transcranial because the monkey skull is much thicker than the mouse. So we decided to use the doing it outside the dura without damaging the brain. So you basically can open a window, a skull window, then you assemble chamber that allow you to do recording, viral injection, but optical stimulations, you put optic fiber outside the dura. You can glue on to the skull, but put optic fiber. So that allow you to do two things. One is without damaging the brain. The second thing, based on your optical fiber, distance from the dura, because of the angle, you can actually activate much bigger brain areas. So as you can see, these are two examples, and you can see you turn the blue lights on, you see activation of neurons, and turn the orange light on, and after a while it will inactivate this, just back to the normal baseline. So this is actually very effective. You can see majority of neurons unit, about over 60, about 60% of the unit can be activated. When we turn the lights on, and about 90% of these activated neurons back to normal when we put orange lights on. So there is a so the activation is quite robust. Average is about the doubled, 100%. Double the magnitude, and the latency is about about 4 to 5 seconds when you turn lights on to the fully activation. And the average thickness of the cortex is in the macaque monkey is about 2 to 3 millimeters, and you can see from these depths, we can actually completely, the activation is maintained very, very well in that depth, even to the 6 millimeter, which is the mouse hypothalamus area, and we can still see a very significant activation. So we think this provides us a new tool who can really facilitate us to facilitate the study of optogenetic applications in bigger brains, especially monkey now, more and more these higher cognitive functions and not only in basic neuroscience, but also in neurodevelopmental psychiatry disorders are being studied in monkey models. We think this will help us to further our study in this area. So another application we think is really important for, especially for my lab and many other labs study autism and neurodevelopmental disorders, that now you can really study during development, very early on, even P0, P5. If you can put a virus in, whether either use a Creline or use a viral injection, it's harder to stick optic fiber chronically during development for mouse models, for any models. But this way, you don't have to. You can just share light from outside the skull, and as I mentioned, larger brain regions, and without the optic fiber, you can do much more complex behavior. And whether this can be in the future use, the clinical is still long way to go and to think about it, but with even more improved sensitivity, maybe the activation of cortical area, prefrontal cortex, and other areas that might be a possibility. So I'm going to stop here. And I want to thank all the people who have done the work. I mentioned the key people in the lab, but I want to mention a couple more things. One is all these studies are large collaborations with many groups. PhD, I mentioned with my classes lab, very close collaboration. The Seoul optogenetics, we collaborate with many labs, Carl Dazros, Ed Boyden, or Miller, Mike Lassa, Zhang Yanfu, and also Guo Changbi and Bob Desmond's lab. And the TRN subnetwork also have many, many collaborators, international and the US, including Portugal, China, and MIT, Broad, and many people contributed to this work. And in particular, I want to thank everyone in my lab who contributed. I couldn't mention them all, and all the supporting staff, all the people in the lab supporting every project and contribute to every project. And finally, I want to acknowledge all the funding agencies or individuals or foundations who really contributed, made it possible. Without their support, it's not possible to do all this research in the lab and to encourage young people think that science is still a wonderful, wonderful career. Thank you.
Okay, thank you very much, Guoping. I see we have a lot of participants. This is way more than we normally have in Building 46. I think we should go into complete shutdown more often and have Zoom seminars. Okay, so now if you would like to ask a question, let me see here. if you would like to ask a question, raise your hand. But I am not seeing where do I see the hands? It's a little unclear to me. Oh, maybe if I see the intent. Here we go. Okay. I don't know who was first because I'm just opening up, but I just see their initials. Whoever has the initial DW, I'm going to allow you to talk. Dongqin Wang, sorry, now I see them. Dongqin Wang, you are allowed to ask your question. No. Is he talking?
No.
No, okay. How about Dara Manoj? I'm going to...
Sorry, Bob, I didn't have a question. I didn't raise my hand. Sorry.
You did or did not raise your hand?
Did not. I don't think I did, but...
Okay, but you're... Okay, so I see your hand. Okay, how about...
Sorry.
This Zoom meeting is not entirely without its flaws. Tsuneya Akitsu. Tsuneya.
I'm sorry. Yeah, thank you. Great talk, Dr.
Thank you.
Dr. Feng. How could you control the firing rate using the Soul technology?
So that's one. It's a very good question. That's one of the advantages of Soul is in general because you don't have precise timing. So it's actually is to depolarize the membrane. So it will follow the increased excitability, basically, more likely to fire. So you basically allow to follow the endogenous firing frequency instead of control the precise frequency yourself. So if you want to precise control, you have to use channel rhodopsin. So in many of the cases, we prefer that because many of studies use 20 hertz, 30 hertz, but the firing rate in vivo, in real neurons are very dynamic. So we think that's in some cases maybe a better way. But in this, if for SS4, you don't control precise your firing rate.
Thank you very much.
Okay, we have Anita Luthi. Oh, allow her to talk. There we go.
You're muted.
Oh, unmute. I'm clicking unmute. And let me just say, is there, no, it should work.
Just a reminder, the people on the other end also have to unmute themselves.
Yeah, maybe you muted yourself. Let's go to...
Is that better now?
Oh, yeah. No,
yes, there we go.
Yes.
Okay.
Okay, so thank you for letting me talk, and thank you for this very nice talk.
Thank you.
Whether you had some more information to provide regarding the expression pattern, the gene expression patterns of the two cell types you pointed out, the SPP plus and the HL1. From all I can see, you say is that it's only the HL1s that express the CAV 3.3 channel. Is that correct?
No. So they are both expressed, but they're differential levels. So the SPP1 cells, which have bursting properties, they express higher the CAV 3.3 and SK2 channels. It's not completely exclusive. It's just differential levels.
Yeah. And any other genes that make them separate, these two cell types?
Yes, there are many genes. Actually, each group contains more than 100 genes are uniquely differentially expressed with a gradient type.
Okay, thank you very much.
Thank you.
Okay, the next hand is Alexander McClanahan, and please unmute yourself. Hello?
Yes.
Okay.
Hey, sorry. Can you hear me?
Yes, now we can.
Sorry about that. Thanks, Dr. Feng, for the talk. The Soul data was really exciting. It makes me think that maybe you could envision a system one day where you could sort of target specific receptive fields with stimulation, which would be really cool for sensory cortices.
Mm-hmm.
But I had a question regarding some of the inflammation data. How long after the injection of the viruses were the inflammation results taken?
So you mean the inflammation thing? I don't remember exactly. Usually we do virus for 3 weeks minimum.
Okay, gotcha.
And we detect, to be fair, so we look at the surface cortical area, cortical top layers, because if there's any damage from the transcranial, they will be on the top. So the picture I show you is on the very surface layer of cortex.
Right, gotcha. Interesting, thanks.
Okay, next is Mark Baer.
Great talk. Wonderful.
Thank you.
Just a quick question, which is whether this SOL method can be used to drive neurons into depolarization block. So it could also be used as a way to shut neurons off.
Yeah, so this is the activation. The only we are trying to develop inhibitory SOL, but we don't have it yet. So our way is we actually can use them for interneuron, excited inhibited interneuron actually can lead to inhibition. That's what we see in the monkey brain actually.
All right, good, very good, thanks.
Okay, next is Shanjun Deng.
Can you hear me?
Yes.
Yes.
Oh, great. That's great talk. Really, thank you so much, Dr. Feng. It's amazing to hear the story. I just have one follow-up question to the single cell data. You picked 2 genes from the SPP1 and ECL1 from the group of the marker genes, right? You said like in each group. How do you pick these 2 genes from among the other genes?
So so these 2 are the more I would say the more almost, if you compare these 2 themselves, not to other genes, they are almost exclusive. They are not overlapping. So we want, when you do in situ to do localization, you don't want them overlapping. It's hard to tell. So these are the 2 genes to us seems like the most non-overlapping.
I see.
So we identify 2 extreme groups. There are many in the middle, in between, like they don't express either, or they a little bit of both, but these are the most separated genes that we can use as a marker.
Okay, great, very cool. Thank you.
Thank you.
Okay, the next question is from Ted Wu.
Hi.
Hi.
Hi. Not very nice talk. Just 2 quick questions. So I'm very interested in this SOL actuator. So I don't know why can you use the right light to stimulate as the light for the stimulation?
Yeah, so that's a good question. We found yes by accident. What we found is if you take, so we have a mouse crossed to excited neuron Cree. We have a conditional express mice. These mice are being sent to Jackson Lab now. So you can inject Cree into any brain regions. So what we found is if when we cross these mice to NEX Cree, which is excited neuron specific Cree, and if you use the loom light, actually they are okay. But if you put in a hood, very strong light, just white light, it will activate, we think it's active retina. Then if you do C4 staining in the visual cortex, you can see a lot of C4 expression. So to answer your question is yes, you can use a white light actually to activate it if it's strong enough.
I mean right light, right? Right light.
What do you mean right light?
Right light, not white light, right light, right.
Okay.
Because the blue light is visible, but
Yeah.
the red light is not visible. It's
Oh, you mean red light?
Yeah, red light.
Oh no, not yet. So we are trying to do red shifting, but we have not completed that yet. We're trying to do if with the red light it's even better. It actually can penetrate deeper. So we're hoping to do that, but we have not. We initiated a little bit, but we stopped. So we haven't succeeded on that yet. It's a very good question.
Okay.
Good point.
Just one quick question. So the SOL is an ion channel or not?
So yeah, so it is the same channel as channel rhodopsin. It's a cation channel, and it just add mutations to add different properties.
Okay. So it's still permeable to calcium, right?
Yeah, it's still permeable to calcium. The most studies show that calcium may not be the keys, but it does have calcium permeability.
I see. Because some studies, including my findings, we find the calcium influx through the optogenetic actuator will actually affect the short-term plasticity. So
totally possible, depending on the neurons. Yeah, it's possible. Yes.
Okay, thank you so much.
Okay, the next question is from Minjung Kim.
Hi, it's always impressive how fast and rapid progression happen in their lab. It is really impressive.
Thank you.
I just have just like a quick question. It's not really scientific related, but more technology related. I wonder whether it can be achievable at some point, the patch-seq technique from the in vivo setting, whether it can be actually achievable.
You mean for in vivo
like
live? So it is possible to do, I believe, especially with avoiding the automatic patch. You can, we have not tried, but theoretically, it might be possible. This is a good question. If you do enough, you may succeed in some of them. My feeling is, yeah, with automatic patch, you can actually, if you patch the cell, then you define how many microns you go deep and you suck. I think it's possible to do. Yeah, it's a good point. Actually, we never thought about it, never tried, but I think it's possible to do.
Okay.
Okay, it looks like the last question is from Ning Liao. Liao. Ning, are you going?
Hi, can you hear me?
Yes.
Hi, Guoping, thanks for that great talk. I have two questions. One is about the SPP1 and the ECE one. Genes, do you think they're actually genes that are driving developmental programs, or do you think they're more like just marker genes?
Very good question. We think these are just marker genes for us. We don't know exactly what is their function in the TRN. They are also in other cells, in other parts of the brain, but in TRN, we use that as a marker. There are evidence from mouse study. We know that the early development, at least SPP1 is not specific. So they are probably not driving development. So we are still in the process of doing developmental studies to see. Ideally, you would do a similar study in a single cell during developmental stages. And Mike Halas and I, this is one of the aims we try to do, but we haven't completed that yet.
Okay. My second question has to do with SOL, because it's also activated by the same wavelength as, say, if you want to image GCAP. Do you think it's actually possible to use it simultaneously with 2P imaging, because it's super sensitive, right? Do you think it'll get active?
Yeah, it's a very good question. We thought about this. Some other people also discussed it with. I think it's really was to do a try 2-photon, because previously 2-photon activating optogenetic was very difficult because of the sensitivity. But with this, it might be totally possible, but we have not tried. I think theoretically, in theory, this should be working better than the ordinary channel option for 2-photon analysis. But the only issue is it's not a fast action, so you cannot do, say, 20 hertz. I want to see a 20 hertz spike of the calcium. So.
Right. Well, it's more of like, if I were to be imaging, will I, I were to be imaging, will I unintentionally activate the channel, perhaps?
Oh, yeah. So that you will probably.
Okay.
But even at the focal point, if you imaging, yeah, it will probably activate some of the channels. Yeah.
Okay. Good to know. Thank you.
Thank you.
Okay. Well, that's the end of the questions. We can all silently clap in our homes for our speaker.
Thank you.
Okay. Thank you, and thank you everyone for attending.
Thank you. Bye.